Thank you for the introduction, Mr. Fischer. What I'd like to do today as an introduction, today's topic is to give you an introduction on what biotech is, what genetic engineering is, and I'll give you a mix of the history of biotech, the techniques that we're able to use now, the applications that we're using them for now as well as in the near future. So I hope that by the end of my talk I will not have utterly confused everyone here, so please bear with me. So what is biotechnology? Basically it's any process that uses a biological system or a living organism to produce useful products. And these can be used for agriculture, so we can genetically engineer crops to produce enhanced crops, for medical purposes, to produce biologics, which can be anything from antibodies to cell therapy to gene therapy, but this doesn't include small molecules. So typically whatever you're swallowing as a pill, so aspirin, ibuprofen, etc., these are small molecules. They're produced with medicinal chemistry and are not produced in a biological system. And then the third bucket is industrial biotech to produce chemicals, textiles, etc. So as you can see, these are pretty high-level descriptions. The applications and techniques are very vast, and thus there's a widespread confusion, I feel, on what it actually encompasses, what the possibilities are, and also what potential risks could be. My expertise is mainly in the medical applications, but I feel that it's important to give a bit of a background on humanity's journey and our origins in biotech. And as Mr. Fischer already mentioned, we've been engaging in biotech since we started settling down thousands of years ago. And then one of the first applications, of course, was to produce wine, beer, etc., with the help of microorganisms. Of course, we didn't know that it was microorganisms that were causing fermentation, that were consuming sugars and producing alcohol in this way. We only later started to appreciate that we were actually using biological systems to produce useful products. And one of the oldest applications of genetic engineering actually is breeding. Breeding animals, breeding plants, because there you're selecting for specific traits and breeding them, so you're actually already modifying the genetic code of these plants and animals without actually knowing what's going to come out of it. And this is nicely exemplified in such crops as bananas and corn, that we've been actually modifying our foods for millennia. Modern day bananas and corn look nothing like they used to look like, and they have, of course, much higher nutritional values and produce more food than we've been able to do. And now, in addition, as already mentioned by my father as well, is that we can also select for traits to reduce the use of pesticide use and insecticide use. And if we look at our journey of when we started breeding to what we're doing now, it's very interesting, because originally breeding, we didn't know exactly what genes we were modifying, so we were modifying many genes at the same time, and then selecting for a desired outcome. A more advanced technique that started to come into place in the 1940s was actually that we subjected the seeds to irradiation or chemicals to induce mutations, but also all across the genomes of these crops. And then we selected again for desirable outcomes. And then only with the advent of us understanding what the genetic code is, we were able to make mutations in a more specific fashion and thus specifically modify single genes in the crops and in a predictable and precise manner. So we've come a long way from these early genetic engineering techniques, and as you can see, all of these modify the genomes of crops, but now we're able to do this in a controlled and precise manner. However, unfortunately, this is not appreciated in the widestream media, and there's a lot of fear on how safe these more precise techniques actually are. So we've come a long way to be able to increase our food output with such technologies, and we will further move ahead as we gain an understanding of more precise understanding of biological processes. One of these examples is cultured meat, where we will be able to produce meat outside of animals. There's, for example, this company, Mosa Meat, where colleagues of mine in the venture fund have actually invested in, and the idea is to produce cow cells outside of the cow in a petri dish. At the moment, it's still pretty expensive and I don't think very edible yet, but we will be getting there. So the same if we look at the biotech journey and us becoming more precise, the same holds true for the medical applications. The first way of actually vaccinating was called variolation. So back then we had huge issues with smallpox, and what people started doing in China and the Ottoman Empire was to actually transfer the pus from smallpox pustules to non-immune patients. They made scratches in the skin of these patients or not yet patients, and thus conferred immunity on them. Of course, then later in the 1800s, we discovered that if you took cowpox and used that as a vaccination, that this would also confer immunity with less dangers. And then moving towards today, now we can specifically kill pathogens or use attenuated versions, or even use just parts of these pathogens, proteins of pathogens, to vaccinate people. So we've come a long way from actually transferring pus of smallpox patients to healthy people. And it's estimated that now vaccination actually prevents the death of 2 to 3 million people per year. We've eradicated smallpox, we've diminished the existence of many other infectious diseases, and people are working on finding more vaccinations, for example, against HIV. HIV is a bit more difficult because it's a highly adaptive virus that mutates quickly, so this is still work in progress. But when first vaccination was introduced, people were a bit afraid, let's say, and while vaccination quickly spread in the early 1800s across Europe, many riots broke out when certain countries made this vaccination compulsory. Liechtenstein was actually an early adopter of mandatory vaccination. Johann I of Liechtenstein introduced mandatory smallpox vaccination, but also here there were many protests across Liechtenstein because people were afraid. And back then this was a new way of doing things. Now we've been familiar with this technique for 200 years. We've eradicated smallpox and many other diseases. So if you look at the current measles crisis, it's interesting. In 2000, measles were actually already considered eliminated from the US. Now, already the first quarter of this year, you have as many new cases as all of last year. And in Europe, you also had, I think, around 80,000 new cases in 2018. And this comes back from a misinformation, a study that was published, I think, around 20 years ago, where somebody claimed a link between the vaccination of mumps, measles, and rubella and the development of autism. This was a study done with 12 children. Since then, this study has been retracted from the scientific journal it was published in. There have been many studies conducted showing that there is no link. The most recent two actually was with more than 1 million children, showing that there is no link. However, this misinformation prevails. Penicillin, of course, was the first antibiotic, so it was a huge breakthrough in medicine. What I would like to highlight here, though, is that when penicillin was first discovered, you couldn't immediately just generate tons of penicillin and give it to people. Actually, the penicillium mold was only able to produce a little bit of penicillin, so we had to breed strains that could produce a lot of penicillin for use. So, like we did with agriculture, we actually introduced random mutations in the penicillium mold so that we could get strains that would produce large amounts of penicillin. If we were to do that today, we would have much more precise techniques available to actually much more efficiently and predictably introduce such mutations to increase the yield. I think the advent of modern biotech actually started with us cracking the genetic code. So how is information inherited in a specific gene that's located in DNA, and us understanding how the DNA that's in the nucleus gets actually transcribed to RNA and then ultimately translated to proteins, which build large parts of our bodies. And with us understanding the genetic code, we could improve our techniques of modifying it. And that led to the advent of recombinant DNA technology. So the first applications were that you could insert human genes into bacteria that then would be able to produce the human proteins. And that's important, for example, in such diseases like diabetes, where certain patients don't produce the insulin protein. So what we used to do is that we took animal insulin. So we would isolate the insulin from the pancreas of pigs and cows and then give that to patients. Of course, now that we can produce human insulin in bacteria in a very efficient manner, is beneficial to patients because it's more effective, it's quicker, it's less expensive, and it's human protein. If you give animal proteins, our immune systems will generate an immune response against foreign proteins. So this is a very traditional modern biotech application that you can produce biologics in huge quantities in cells in bioreactors. And as our recombinant DNA technologies have evolved, we can now use them actually for human therapy. And I would like to highlight two particular techniques here. One are viral vectors, and the other is the more recent CRISPR-Cas9 that you've probably heard of. And as is the case with biotech in general and biology in general, is we discover a process a biological process, we start to understand it, and we start to adapt it for our uses, so medical or agricultural, etc. And here, for viral vectors, we've copied viruses. So what viruses do, okay, let's take a step back. So viruses don't produce their own proteins. So they need to infect cells and use the cell's machinery to produce their proteins and to multiply. So we've used that. We've used viruses. We've removed their viral end genes. We've removed their ability to replicate, and we insert the genes that we're interested in. And in that way, if we infect cells or we transduce cells with these viral vectors, we can insert the gene that we're interested in. And the example that's given here is that you have sickle cell disease, where patients have a defective beta-globin gene. So you use the viral vectors to include a working beta-globin gene into the red blood cells, into the stem cells, and then you put them back into the patients, and like this they can produce normal red blood cells. So this is a very, very efficient and effective way of introducing the genes that we're interested in. Then one of the most recent discoveries was CRISPR-Cas9, which was actually only discovered in 2012. And this is another biological process that actually bacteria use to defend against viruses. So what this complex does, the CRISPR complex is able to recognize specific DNA sequences, and the Cas9 protein then cuts at these sequences. So we can determine and predict where in the genome we're going to make a cut and where we can remove a gene or insert a gene of interest in a very predictable and precise and actually also very cheap manner. So that is the advantage of CRISPR versus viral vectors, that we can actually determine where we're going to put a gene. Viral vectors have other advantages in that they can directly move into the cell, while CRISPR needs another packaging system to get into cells. So if you look at these techniques and compare them to random mutagenesis with irradiation, I think we've come a long way. And why is precision important? It's important. I would like to give you an example here of why it's important. So the first gene therapy trial actually happened in 2000. And this was for a disease called called X-linked severe combined immunodeficiency, and it's also called the bubble boy disease. Basically, it's usually boys. They don't have a functioning immune system, so they have to be kept in isolation because they cannot fight any infection. So they have a defective gene called IL-2RG, and the first therapy trial used a gamma retroviral vector to transfer this gene to the stem cells of these boys. This was a major step for gene therapy because it actually restored immunity. However, there was a setback in that these children, or some of them, developed cancer. And that was because these viral vectors actually included the gene in places of the genome that disrupted a normal process, which then caused cancer. So since then, we've improved viral vectors, we've generated techniques to assess where in the genome these genes get integrated. And now, just a month ago, there was published a paper on a new clinical trial where they used a different viral vector to treat these kids. And they've been followed up for 2 years. They haven't developed cancer, and they have a functioning immune system and have been released from hospital. So I think this is it's on one hand an example of why we need precise techniques, and on the other hand, it shows what we can do with these gene therapies. And while such genetic diseases are, of course, very amenable to gene therapy because you know there is a gene that is mutated, it causes a disease, so you replace it with a functioning gene. But there's other applications, and actually the first gene therapy application that was approved in the West was for cancer. And here you take the patient's immune cells, you modify them to be able to recognize cancer, and then you put them back into the patients. And there we've seen remarkable efficacies in blood cancers where we haven't been able to see any responses with other treatments so far. And both of these techniques are called ex vivo gene therapy because you're modifying cells outside of the patient's body versus directly into the patient's body. But so this happened in 2017, and another breakthrough at the end of 2017 was actually the first approval of an in vivo gene therapy for a rare form of inherited blindness. So Luxturna, it's been approved in 2017, and there you subretinally inject the viral vector with the functioning gene, and these patients, you should probably Google this because if you can see videos of the patients at the beginning of the trial and at the end of the trial and how improved their vision is. And this is something where we've never had any kind of treatment before. And if you're, of course, these are rare diseases, but if you're a patient that cannot properly see, this is pretty big thing. There's more gene therapies in the clinic, and we were expecting an approval in blood disease called beta thalassemia this year. There will be further indications moving into the clinic, like Duchenne's muscular dystrophy, and these are all devastating diseases where we haven't been able to do anything ever before. All of what I've talked about before, these gene therapies, they're called somatic cell gene therapies because they actually modify only a specific cell type within a patient. So these are not modifications that will be passed on to the next generations. What is more controversial is germline editing. So there, the idea is that you modify egg cells or sperm cells or embryos, and these are, of course, then modifications that will be passed on to the next generations. We're not there yet. The scientific community agrees that we need to understand this better. We haven't been, well, there has been progress. There has been a study published in 2017 where scientists has been able to modify embryo cells until the blastocyst stage and gotten uniform modification in all of them. Of course, it's not allowed to develop this further, so this was stopped, and there's also some discussion on actually the outcome of this study. But I just wanted to highlight that things are happening, but we're not there yet, and the scientific community agrees that we need to understand all of these techniques even better for such an application. And the benefit here, of course, could be in the area of genetic correction, because you could modify an embryo and thus prevent a disease ever developing in this patient. Here, the idea is that you correct a rare mutation to the genetic sequence that most humans have. This is in contrast to genetic enhancement, where you would introduce a new gene or enhance genes that are not there for an immediate medical need, but rather for enhancement. And this is, of course, much more controversial, and this is what people are afraid of when they talk about this in the media. However, in the West, this is unthinkable that regulators would approve such a study without any medical need. And in addition, you may argue, yes, okay, this is the West, what about other countries? But we're not there yet. We don't understand yet how complex traits are encoded, and such things like intelligence. There's probably no gene for intelligence. It's a mix of different genes, of epigenetic factors, and of the environment. And thus, there's this premature fear is somewhat premature. And just as a conclusion, I've outlined a little bit the journey we've been always engaging in genetic engineering. We have now these techniques at our disposal that are more precise and predictable than ever. We should be taking advantage of it. We can enhance crop yields and nutritional quality. For medical applications, we're able to treat diseases we've never been able to do anything about before. And it's important to have this science-based dialogue to prevent other examples such as the measles crisis we are currently having, which could be prevented by having a more informed discussion. And just as a summary, I think this is an exciting time for us. We now understand much better what we are doing, how biological systems work, and have the techniques to treat diseases and malnutrition like never before. Thank you.